I want to invite Dr. Cobbs as our next speaker. I don't actually have the schedule in front of me, but I think he is speaking about immunotherapy. And Dr. Cobbs is our wonderful tumor surgeon, leads his own laboratory downstairs, has done extensive work in the field of immunotherapy, specifically relating to glioblastoma, is responsible for the discovery that there are I don't know if you'll talk about this today, that there are traces or there are proteins that are at least similar to the proteins found on a certain virus called cytomegalovirus in glioblastomas, and has worked with that in a number of forms, including a clinical trial that came out of that work and was run at this institution, and there's somebody here that was on that trial. So thank you for everything you've done, and welcome.
Well, thank you guys, and I'm really happy to be here and talking to you all. What I will say is that I'm really passionate about glioblastoma research and treatment. I spent the weekend, it was rough, I admit, but there was a philanthropist and some researchers in Palm Springs that invited me last weekend, and I was down there. A bunch of the people I trained with at UCSF were there, trying to get people to move the needle, because I went through my training in the 90s, and sadly there has not been a whole lot to be too proud about in terms of the improvement in glioblastoma from clinical trials over the last 30 years. There have been improvements. Optune is one, but you could count them on, a single hand. And what I want to talk about now is something that I'm very passionate about, and it, in my mind, and I may be crazy, but I truly believe that this has the potential to be something that not only will move the needle a little bit, but literally transform the field. And that's immunotherapy. And you've all heard about immunotherapy for other cancers. I'll just sum up the reason glioblastoma doesn't work in that type of. Is this the talk? Yeah. So let me just make sure I have the slide that I thought I did. Okay. Thought I had another slide first. So the concept for cancer immunotherapy that took over and has been so beneficial for certain cancers like melanoma and lung cancer is that the tumors have so many mutations that all you have to do is sort of turn up the volume of your immune system and they will start attacking the tumor. Glioblastoma is what we call a cold tumor. And by that, what I mean is if you look at the blue cell, it's a glioblastoma cell, there are all kind of molecules expressed all over these cells that try to shut down the immune system. So when your immune system tries to attack a glioblastoma, it literally is impenetrable. The immune system cells cannot get into the tumor. And in the last 5 or 10 years, there have been some very interesting concepts that have arisen that that open the possibility that we can manipulate the immune system by understanding the fundamental properties of how it works in a way that may be able to overcome that so-called cold environment. This is a very basic schematic of how the immune system works, but I think it's important to show because there are two parts of the immune system. There's the innate immune system and the adaptive immune system. If you get an infection in your arm, like a bacterial infection, immediately things swell up and you get redness and pus and everything. That's called the innate immune system. That's an immune system that occurs within hours, and that's programmed through millions of years of evolution. And these cells come in and they bust up things, and then they take that information back to your lymph nodes, and they tell your lymph nodes to make other cells like the T cells and the B cells that are extraordinarily potent and can come back with extremely high specificity and wipe out the tumor or the whatever infection is there. But what happens in tumors is over time, the innate immune system loses its ability to function, and these T cells and other cells that normally are in this anti-tumor immune environment, they become sort of burned out and they become exhausted, and they sort of actually can start producing factors that make the immune system not work. So it's become apparent that there are ways you can actually sort of backtrack and reboot the immune system. That's what I'm going to talk about. So this is just a schematic. Does this thing have a pointer? I think it is. This is just a schematic that, that's not working. Anyway, basically the concept here is that the tumor is here. It's got all these cells in here that are immunosuppressive. Is that a pointer?
Yeah.
Oh,
I think it's
have to be plugged in. Okay. Do I point to that? Oh. Oh, well, I'll just point to it. Oh, actually, the mouse works. So basically, the tumor is a big blob of tumor cells plus immune cells that are actually counterproductive. They're making the tumor have an immunosuppressed environment. But we can activate that innate immune system by putting in things that make this tumor environment think there's a viral infection. And that can cause rebooting of this innate immune system. And if we do that in a way that overcomes the immunosuppression, we can start getting all those other potent immune cells to come back into the tumor, and they can think there's a viral infection. And then when they get there, they'll say, oh my gosh, there's also a tumor here, and that can start getting rid of the tumor itself. So we took advantage of some work we were doing in the lab where we were taking glioblastomas, taking the tumors out and putting them in a dish and looking for immune cells and activation of immune cells. So we can take these tumors out, slice them, and keep the tumors alive in the lab as a big chunk or slice for several days. And then we can stain them to see if there are these immunosuppressive T cells, for instance. And in this slide, you can see these dark cells are the rare the rare T cells that are anti-tumor, and basically they don't even penetrate the tumor, but they're in the bloodstream. They just can't get into the tumor. So in working with a local biotech company here in Seattle, they've developed a product that's based on the RNA vaccine technology where we could potentially inject into the tumor something that will reboot this innate immune system. So we've been working with them, and in our first tests we did, we had them inject something just as a marker that would make the cells turn fluorescent green, and it was pretty clear that if we injected these tumors, we could get dispersion of this RNA molecule, and all these tumor cells would start producing this substance that turned them green, and so we could, as proof of principle, show that it works. So the next step we did, and a lot of this has been in collaboration with people that work in our lab right downstairs, is we took tumor tissue from patients, and we put it in the dish, and we then gave this drug, HDT-401, which activates the innate immune system. And as Dr. Bonham said, my lab group over the years has been studying a viral infection that we believe is associated with these tumors. And when you get an infection with a virus, not only do you establish immunity in the short term, but you establish long-term immunity. And this virus is called cytomegalovirus. And for unknown reasons, it creates a massive immune response and a massive long-term immunity, such that like all of your immune cells, maybe 10, 20, 30% of all of them in your body are focused on this virus. So we, based on some work in the literature, came up with the concept, what if we reboot the immune system by putting this molecule that turns on the innate immune system, and then we put in bits and pieces of the virus to make the immune system think there's an active viral infection into the tumor, and then add the patient's white blood cells back and see what happens. And so we got this up and going in a dish in the lab. And when we do that in the lab where we put the molecule, the RNA sort of the RNA vaccine molecule that turns on the immune response, plus bits and pieces of the CMV virus, plus the patient's immune cells, what we've encountered is that we get a dramatic activation of some factors like interferon gamma, TNF, IL-6, IL-12. These are factors that are critical for driving an immune response to the tumor. So we're at a point now where we believe if we can develop a clinical trial in patients and we can put in this RNA vaccine technology and activate an immune response with the vaccine type of therapy to the CMV virus, we can cause a real profound activation of an immune response that will cause those cells to come into the tumor thinking there's a viral infection, kill the tumor cells, and then the off-target effects of that would be killing more tumor cells, we believe. So there have been studies by some famous people just recently at the National Cancer Institute using mouse models that suggest that this actually may be a way of overcoming this cold tumor environment and activating a new immune response by tricking the body into thinking there's a viral infection in the tumor and activating the innate immune system. So that's pretty much what I wanted to tell you about, that we are, hoping to get a clinical trial going in the next few years, and we've gotten some funding to start the ball rolling. And I, my goal of having been doing this for many years is to push the envelope, get things into clinical trials, get things into patients, which we think will actually change the paradigm and hopefully not move the needle this much, but literally cause a profound improvement in the ability to attack these tumors. And couldn't have done this without a huge number of people, including Parvi Hoti, who's back here, who runs the lab, and all of my colleagues who've been working on this in the lab, and some foundation support from the Susan McGregor Foundation and Paul Allen Research Award that we have to do this. So be happy to answer any questions. Hopefully that wasn't too discombobulated. All right. No questions. Good.